All right, everyone.
Real quick announcement for folks in 501 and 502. 501 and 502, you had your labs last week. The lab reports are due this week. Obviously, I will be in the lab on Monday and Tuesday. So you have a choice of where to drop it off. You could bring it to the lab, and you all know where that is because you were there already. Or if that's not convenient for you, because I will obviously be taking a lunch hour too, and so I won't be there the whole time as last week. You can just bring it to my mailbox at GFL, third floor. Just drop it off at my mailbox. That's fine. Don't forget to upload it too. Remember, hard copy and soft copy. So use it, bring it to the lab, or what might be more convenient, just drop it off at my mailbox on Thursday, tomorrow and Thursday, depending on what section you're in. Yes. What is my lab? 503 lab starts at 11:30. 504 starts at 10:30.
So for my students, I will bring your prospectuses with my feedback on them to the lab, so that you will then be able to use that in your working of the lab report. When you turn them in, we have discussion I believe we have discussion next week, so you can simply turn in the hard copies to me in discussion for my students and upload them.
For 501, the lab reports are due tomorrow.
Tomorrow, yeah, 501 tomorrow. Yeah.
We have a week from our lab.
A week from the lab, yes.
1 week from the last.
Yeah.
Okay, thank you.
Yeah.
All right, I'm just going to finish passing back the rest of the homeworks, and then we'll get started. So if you want to still chat her quietly to yourselves. Megan, both of you, there you are. Should be about another minute or so. Luca, oh, you're in a different place today. Isabel? Christian? Congrats. Ryan? Merselbard. Azam? No. Are you in his group? Jack? Amalia? Okay. Thank you. Ryan Scott? Alex Soka? Sarah? Truman? Harna? Oh. Make you all sit down. Thaddeus? Last call for Ashley and Tom. Okay. Oh, great.
Professor Bell?
Yes. Oh, wow. Send me an email and yeah, I'll fix that right up. You'd be surprised how dumb I can be. So, yeah. Well, you'd think I'd see it, but yeah, just send me an email and I'll remember that. Okay, everyone. We'll be going over the homeworks here in a bit, but if you wouldn't mind kind of just putting those down for half a second. Last night in Las Vegas, Nevada, there was a tragedy of sorts. The largest mass shooting in the history of America has just taken place. This is actually where I grew up, Las Vegas, so it takes a particular toll on me. So I'm not usually one forgiven for prayers, but if you wouldn't mind just joining me in like 30 seconds to just kind of quiet in the room, I'd appreciate that. So. Thank you very much for that. Now you can pick those homeworks back up, and we can kind of review them. All right. So this is the sort of class where I'm not going to be giving you the answer key. I feel it helps the next generation more than they helped you. So what we'll do is actually just go over all our homework assignments, test together here in class so that we all understand what we're doing. So the first question, you basically got some sequence. You gave it to me while you were asleep. It's very weird. But then I asked for the first question, sort of what was meant by the 5' and 3', and I was basically looking for three things. And if you missed any one of those things, I just took a point off. So if you see something like directionality of RNA -1, or you didn't have a hydroxyl or phosphate group -1, that's sort of where it comes from. But you basically needed to know that they indicate something about DNA, that they related to the sugar phosphate backbone. You needed to have some sense of where the phosphate and the hydroxyl groups were. And you should know that it basically has to do with the directionality of DNA. That they go anti-parallel, that they polymerize in a certain direction, that they do something of that sort. This is just the basics that you need to know about the 3', 5' stuff. And I'm going to go a little quick, so this I don't expect you to all write everything down. I just kind of want you to know this. This isn't important to have in your notes. It's important to have in your head. So. Another thing I'll be doing as we go along is I'll tell you how many points that I, as a professor, lost the class by not teaching this material well enough or by not being clear enough in what I was asking you for. So I've lost us 54 points with this very question. Luckily for the second question, I only lost us 11 points. This was basically just match up, convert from DNA, from the coding strand to the template strand, and make sure you get the primes right. Most of the errors here were just someone wrote a G when it should have been a C, or they didn't get a U, or popped in a U there every now and then. So real simple stuff there. Most of you, I think, have a good grasp of the complementary of DNA. So then I said, okay, now that you got this, sorry, was there a question? Nope, okay. I said, basically, once you got that strand, tell me what the mRNA from that strand would be. So it's just another complement, and it should be the same as the first strand with U's instead of T's. Again, we didn't lose too many points here. It was mostly just a transcription error. Every now and then someone had a point mutation or an insertion or a deletion. No big deal. I think you all have a good grasp of that. However, we lost a bunch here. This was actually our second largest loss. And it's because it is deceptively simple. I think if you just go for amount written to points you could have achieved, if you had literally written a G there, you would have gotten full points. And you could have written entire sentences and paragraphs that were wrong. The entire point here is that when you're basically making the mRNA, when the RNA polymerase goes from that DNA strand that we had worked with to make this mRNA, what's the first thing it puts in? Well, it always polymerizes 5' to 3'. It always reads from 3' to 5'. So basically we just go look at our first nucleotide, in this case, just guanine. Simple as that. But since it was basically an all or nothing question, we lost a few good points. The final question on question 1, basically you needed to use this little codon table. I've corrected this little U. Not on your homeworks, unfortunately, but just in all the stuff you have now. But most people got this. Usually it would be you missed one protein here or there, or you happen to have substituted one for another because you just didn't look it right, and it's 1 a.m. and you're bleary-eyed anyway. When I had mentioned the letters to use, this is sort of what I meant. Just these little letters that accompany it. If you put down the actual 3-letter abbreviations, that was perfectly fine too. It just made it harder to grade. Because I can read that really quickly. It's like Gibby, Sitzy, Slithloq, Lenny C, N. So I got pretty good at just saying those things. Any questions to that point? That's just question 1. And that was pretty straightforward, and I think most of you, I think you all had a good grasp of that one. All right, question 2. This one was a bit trickier. It's basically the same thing as question 1, but a little longer of a sequence and starting with the mRNA instead of the DNA. So you didn't have to do any of that DNA stuff. First thing I wanted you to do is give me the codons. So the first few codons, the first anti-codons will basically match up to the first 4 that we've got up here. this should be represented 3' to 5'. You know, you can kind of do your mental gymnastics thank you, everyone, to sort of figure out how that would all work. And this is the result you get. You get AAA, CAA, you get it. And I actually gave, very few people actually separated out these sequences. Usually you just do 3' to 5'. But since these are all on separate tRNAs, yes?
Is there no 5' after the
Oh, look at that, I got an error. All right, well, for that, I'll give everyone 1 point. You all, that's, see, it's good to point out my mistakes. That is, if I can't be held to the standard, who can? All right, so you didn't lose quite that much, but we lost a bit, usually with either the 3' or the 5' not being in the right direction or unlabeled. And some people just chose entirely different anti-codons. So, yeah, that was a big one. Any questions on why that would be the answer or why it wouldn't be the answer? Okay, I will continue. So question 2b, that's basically, let's go back to this table, do the same thing again. This one was 5NK, Lexia, Laville, Erg. Simple as that. Most people did pretty good on this. Actually, I'd have to go back. I think more people got this one right than the... No, it was about even. If you missed one. And usually, again, the errors were just, you got one of the protein, or one of the amino acids wrong. Yes? Was there a reason I separated them? Oh, so it's easier for you to read? Yeah, it's... Otherwise, you've got 30 letters you're looking at, and it's just kind of random. So you'll actually notice while I was looking at it, I probably put a little dash here and there. That was just so I can read it. Yeah, literally no other reason. Merely typographical. Question 2c, this was actually the biggest one. And that was because I was looking for very specific information about the final protein. I was looking for some information just for you to show some of your work on what would happen to the amino acids, or to the to the nucleotides. I was looking for a very specific kind of mutation. So some people said it was an insertion or a deletion. But you actually don't know that. A lot of people tried to sneak by substitution mutation, but that's definitionally what it is because I changed it. I was looking for the silent, nonsense, and missense. So a silent mutation, what happens here is this remains the same amino acid, so the final protein, nothing happens. With the nonsense mutation, that stop codon says stop coding all these amino acids, and so the final protein is cut short. Some people said it gets damaged, or it's incomplete, or it's there's a number of different ways of saying that protein changed in a dramatic way. And then finally, this missense mutation is when we now substitute one of our amino acids for another amino acid, but the final protein itself might change in some way. And that's what I was looking for, not just what would happen to the amino acid, but what would happen to the final protein. So in this case, you could just say, the tertiary structure might change, or it might do something. I was just looking for any reference to the final protein not being exactly the same. And that was all I was looking for. This was sort of where our biggest one was. And this just allows me to point out here that an amino acid is not a protein. A protein is made of an amino acid. Amino acids comprise proteins. Actually, I suppose comprise works both ways in that. Amino acids go into the making of a protein. So this was our big one. I apologize for that. And actually, I think one student actually pointed out, quite rightly, this one right here codes for a cysteine, and there's actually for what we're about to reveal is, there's two proteins that formed a complex. This is actually the cysteine molecule that's part of the disulfide bond between the two proteins, amino acid sequences, and therefore the insulin itself would just kind of come apart. For that, I think you got like 2 extra points. So, way to go above and beyond. This was a pretty easy one. I was mostly just looking for something like this. If you want to quibble about a point here or there, I'm willing to hear that. But basically, I just wanted you to know that a ribosome has 3 parts. It's got 3 sites. That when they bond, that that you don't bond to the tRNA and the amino acid sequence at the same time, in that 3 is not connected to its tRNA anymore. That was kind of where a lot of the errors came from. A lot of people drew the mRNA, which was really good, but basically something like this. And just so you're familiar with what ribosomes look like and act like. Any questions on this one? On question 2? I think this was most people. Yes.
What's the dihydrogen nitrate? Or what is it at the start?
This little thing, I believe that's from the methylation. So that's a lot of people recognize that there's a methionine that gets put at the top and sometimes cleaved off. That's not in this case. Does that make sense? You didn't need that. I didn't care about that. It is basically, we're hiding a lot of the complex chemistry from you, and just how do any of these things actually bond? Well, there's a group available for it. That's something you'll learn in your biochemistry class. For us, we're just going to hand wave it and say, don't worry too much about it. Is that? Yeah. No one lost any points for including that or not including that. I didn't. Yeah. Okay. Good question. All right, this final one, or not this final one, I basically said, go look at this tool and try to use it. I gave you very little in the way of instructions on how to do it. But we tended to stumble and bumble through it, so we had this amino acid from our first part, and I said, knowing this isoelectric point, knowing this molecular weight, what is this protein? Well, if you go to this website, and kind of looks a little bit like this, you input the isoelectric point, the molecular weight. I also gave you a hint that you could have put something like Homo sapiens or human beings there. That would really help narrow down your search. And then you just put the ratio of what each of these things are as a percent. That threw a few people off, but seemed that some people, you eventually got it. But yeah, that's what this says right here. Do it in a percent. So you should have gotten back an email that looks something like this. It gives you a sense of what your amino acid composition is. And then you get basically 3 charts. 3 charts are sent to you. First one kind of looks like this. In this case, it'll either say for the species Homo sapiens or for all, depending on what you do. And that top ranking result says insulin A chain. That's pretty great. If it now does it for any species, you see that you actually get a whole bunch of insulin A chains. For cows and maybe a chameleon or something. I don't know what Gorgo means. But basically, what this will allow you to see is that the amino acid sequence for the insulin A chain is actually retained in a lot of mammals and a lot of organisms, because nature, once it figures out how something works, tends not to want to break it. So, and then the third chart you'd get would basically say, okay, if I just restricted this to human beings, what would it look like? Well, it would look like insulin chain A, maybe some endothelial thing, some intermediary protein, whatever. But again, at the top of all these results is insulin chain A. So you basically write down that result, insulin A chain, 3.79, and the molecular weight. These two don't really matter. I was basically showing you that the initial ones I gave you were close. Good guessing. Okay, so you do the exact same rigmarole for the second part. So you get the same composition. You get in this case, insulin B chain. You get it in all 3 graphs, just where you'd expect to get them, and you write that down. Okay, so then this next question, I think very few people lost. I don't know where these went. Very few people lost any points on this. Maybe a point here or there, but basically makes insulin. You all figured that out, or we figured it out. And then this final question was just name any disorder. I basically wanted you to do a little research and figure out how is insulin used in the body and how can it go wrong. Bonus points to those that said anything other than diabetes. For going we all knew diabetes, so if you just chose literally anything else, you would have gotten a couple points. So in this class, it will always pay to be creative. So yeah, find something unique and tell me all about it. Okay, so for this final question, a lot of you had heard me, at least at various points, that this was something of a gimme question. This was basically me as an instructor extending you 10% of your grade in this homework assignment to just tell me how you think, how you approach a problem. Especially one that's open-ended and one that is not Googleable. But that didn't stop any of you from actually taking the question and almost Googling it verbatim. So if you take the exact question, it turns out every single one of these sites got listed by us collectively, and that's okay. Basically means I know you all turn to Google first, and turning to Google first is actually okay. This is a good way of doing, casting a wide net and figuring out kind of what that surface level knowledge is. Then I think almost without exception, very few of you chose anything other than the genome.gov. And this is to let you know that I have in fact scanned in all your papers. But yeah, so this is all your answers. I was actually really surprised to see that a lot of people said they trusted the government. That is, maybe that's a generational shift thing. When I grew up, we didn't trust the government. Another thing I found, but you should trust genes. That's a good point to discuss here. Is you'll actually hear a lot of debates, especially with, say, the current administration, in regards to a lot of scientific issues like climate change. This was true in, say, the Bush administration for stem cells, and the suppression of information that we as scientists have. So the fact that you all went looking for basically government veracity of what we know to be true speaks to the importance of our government being honest and true itself. So always try to keep them honest and keep them working for you. Then I also saw a surprising amount from a site called Jupiter Scientific, which was something I had never seen. So I went to this site and it looked a little bit like this. Which, you know, that doesn't mean it's a bad thing or anything, so I decided to check out the homepage. Well, this should be a bit of a clue to you that maybe this isn't the best information in the world. And if that isn't, they got a little, just so you know, this website has been bestowed with the following award by Zesta logo, a brilliant design. So, I didn't want to judge a website that hadn't updated its HTML since 2003, which was, in fact, found in the About section. So I said, oh, you know what? I'll check out their scientific books and publications. Well, it turns out the first book that they had was a thing called 1, 2, 3. This book is for me, by the website's owner, Stuart Samuel. The book begins, as all great classics do, 1, 1, 1, how much is 1? 1 is a sugar-covered hot cross bun. 1 is also more than none. This book is fun, he says parenthetically. I do not kid you, this is a real published book from the site you cited. All of this to say, be careful. Because it turns out that other site, that exact same site, told these same people that somehow 8 or 9% of the nucleotides had yet to be determined. Would anyone who wrote down this answer explain what they mean by that? Yeah, that's okay. It doesn't make any sense. We have sequenced the human genome. You just cited the government website that said we have completed. We know what all the A's, G's, C's, and T's are. So this is a dubious claim taken from a dubious site that you really ought to have thought maybe a little more critically about. So just in the future, if something doesn't seem like it would make sense, it might not. And then a lot of you also cited .orgs as a reason to trust something. So here's me just finding out that I could buy a website called humangenomeproject.org and tricking many students for the bargain basement price of $250. Well, it turns out that's a little too rich for my blood. What if I just made this website up? I can get that for $8. It turns out I can also get Jupiter Science for $8. And just in case someone tries to say that's the fake one, I can also get the real Jupiter Science also for $8. So all of this to say, I know a lot of you kind of cited .gov and .orgs as reasons to trust a website. Don't let that be your reason. Let there be better reasons for you believing these things. As for the base pair results, it looks like we settled around 3 billion, most of us, with just some small chunk of us saying about 3.3, 3.2, 3.5, and they're in between. The number of genes we tended to find between 19,000, 20,000, 2,500, 21, 2,400, 30,000 was a pretty good number we all found. 35, and then one said 100,000. That's actually a bit of an outdated number. Now to show you how I did it, there's an actual database for these things called the Ensemble database. It's from Europe. It's from the European Bioinformatics Central. And this is their human genome. And you can find out the number of base pairs for every single chromosome, the number of variations, the number of protein coding genes. You can basically do a lot of things. And what they found is it's about 3.08 billion genes with about 150 million variants. So you could get up to potentially 3.2 billion. And these code for currently known about 20,412 protein coding genes. Now, if you throw in the rest of these, you might get up to about 22,000. So that's kind of the way I found the number of those things. Another thing, I was kind of surprised that no one would try this, but you can actually download human genomes. Like you can actually just go and get them off the internet. So one of the tools you can use to do that is just a thing called Emboss. This is what I had used to do it. Went and found myself a place where they store these things. I unzipped it, and then I ran a sequence for every single chromosome. And this is sort of the GC content. Oh, and I should mention, they just had a little function that said GC, so I said, why not use it? G's and C's will be equal, so that means I'll also know A's and T's. So I just found the GC content for every single chromosome, which in this case amounted to about 41.6% in the total genome. That meant AT was equal to 58.4, which meant that GC, A, and T were about 20.8 and 29.2%. So that was just a way you could have proven it to yourself without having to trust anyone else for your answer. All you'd have to do is trust that they provided you the actual correct human genome. But just in case they didn't, the techniques we're about to talk about would give you the same information. Any questions to that point? Okay, well then I'll make one last point that I trust you all to do your homework. I don't want to belabor this point, but you may ask each other how you come to do things, but you may not ask each other how you did them. Does that make sense? The only reason I'm pointing this and kind of emphasizing this here is don't swindle yourself out of a good education, and an honest one. I'm going to trust you to do that. All right, we're going to take about a 5-minute break, and then we'll come back. Yes. No, that's fine. I'll make a point of that. The only reason we had mentioned to do it on campus is just in case I lost that. You can. Is that a place for assignments generically? Yeah. Okay. So you should get in the habit. I'll make an announcement of that. Definitely just submit it online, but I'm not grading online. It's more there for your benefit, so that you are covered in case I lose it somehow. I'm going to try not to lose it, but just in case you've basically got an electronic backup.
So at this point, there's really
no point. Yeah, I mean, you can if you want, but
it doesn't mean.
Yeah.
Okay, thank you.
Yes.
Sorry, the way I got this was like actually that's how I got it.
Yeah.
Even though I see them.
Yeah, I think you still. I think it's still.
Okay, yeah, I saw the.
Yeah, I mean, I think I figured that out. No, actually. Yeah, okay, that's a good enough reason. Oh yeah, that was what I wanted you to say anyway. Yeah, and that's what I figured. I just needed to make sure. Let me. I don't know where I put my pen. No. Looks like it, but no. Give me that right here. Honestly, we'll always pay. Email me. Thank you very much. Now, are you reading that before the movie or after? Okay, so before. Scary? Are you spooked out?
It's like atmospheric.
All right.
I don't get scared.
Okay. Well, did you see the recent announcement I made?
Yeah, I saw The Shining.
Hey, you should watch all of those. Those are excellent horror movies. They'll spook you out. There's few that you can like Halloween or like Jason or Friday the 13th, Hellraiser, Nightmare on Elm Street. Well, yeah.
The first and second were good, and the third was just me.
Well, isn't the third one where they go off and do like an entirely different thing? Where it's like, yeah, it's like it's the weird mask thing with like the toy maker or something. Yeah. I don't understand that movie at all. It's weird. No one has ever seen Halloween 3. I think we may be the only people in the university. Halloween 3 fans? Halloween? No, it's okay. You are not missing out. All right, everyone, we're gonna come together. I think we're just missing a couple of people. In sort of solidarity and getting past our homeworks and just getting on with our days. Our 3 quotes today are going to be a little tangential to our topic, but they're going to be about resuming the struggle. So this first one, where I actually get the sort of the phrase from, comes from Samuel Beckett in his just the greatest play of all time called Waiting for Godot. If you have not watched it, watch it. The first act is the same as the second, and it's brilliant. Watch it. It begins, nothing to be done. Actually, would anyone like to read it? I don't have to read it. No? Okay.
Nothing to be done. I'm beginning to come around to the... Oh, wait, one second. I'm beginning to come around to that opinion. All my life I've tried to put in saying that me or be reasonable. You haven't yet tried everything. And I resumed the struggle.
So this is 2 people who are waiting for a person named Godot. And haven't been down on their luck. They have tried to do everything. But they've continued at it. And we will too. Thank you very much. I'll read this next one. This actually comes from Theodore Roosevelt in his speech, Citizenship in a Republic, our former president. It is not the critic who counts. So as you did your homework, recognize it's not me that matters. It is all of you. It is not the critic who counts, not the man who points out how the strong man stumbles or where the doer of deeds could have done them better. The credit belongs to the man who is actually in the arena, whose face is marred by dust and sweat and blood, who strives valiantly, who errs, who comes short again and again, and because there is no effort without error and shortcoming, but who does not actually strive to do the deeds, who knows great enthusiasms, the great devotions, who spends himself in a worthy cause, who at best knows in the end the triumph of high achievement, and who at the worst, if he fails, at least fails while daring greatly, so that his place shall never be with those cold and timid souls who know neither victory or defeat. The fact that you are all trying is what matters. So continue at it, and long for the days where presidents spoke like this. I apologize. I will not politicize this classroom. And then finally, the last one comes from Ayaan Hirsi Ali, a famous Dutch parliamentarian, originally from Somalia. She's an immigrant. I believe she's now in the United States. This is actually from her book about her journey from the Netherlands to the United States called Nomad. Would anyone like to read this one? How about Beck?
The trap of resentment is probably the worst mental prison in the world. Is the inability to let go of anger and perceived or real injustice we suffer. Some people let one or two, or maybe 10 unpleasant experiences sway through the rest of their lives. They let their anger ferment into their personality. They end up seeing themselves as victims of their parents, teachers, their peers, and preachers.
Thank you very much. All this to say, don't let losing a point here or there matter in the grand scheme. Don't let bad experiences poison you. Do not become resentful. We will resume the struggle by beginning lecture number 6. Sequencing techniques, number 2. And we're actually going to be talking about next generation sequencing techniques. Now, what do I mean and what is typically meant by next generation sequencing? Recall that I had shown you this graph showing the price of the human genome per time. When we're talking about the next generation, we're talking about those technologies that have enabled these catastrophic changes, these just immense differences in the price, the time required to sequence genomes, to sequence our genetic material. So that's what we're going to be talking about when we talk about the next generation. And today we're going to be talking about 5 different kinds of next generation. And these are sort of the main 5. There are others and there's lots of variations, but we'll basically, for our purposes here, consider them the 5. So there is sequencing by ligation. This is sort of the sequencing by ligation. We've got sequencing by synthesis. This is when we had talked about Illumina last time. That was sort of what we were referring to. You know, why do teachers ever choose to use yellow on white? It's terrible. Ion torrent sequencing, also known as ion semiconductor sequencing. We'll get into that. Is the 3rd. The 4th is SMRT sequencing. So that is single molecule real-time sequencing. You don't need to write exactly single molecule real-time. I'll give you a chance to write that later. I'm more introducing the material here. And finally, there's pyro sequencing. And this was made famous by Roche with their 454 machine. We'll,
Does it have anything to do with fire? That's what pyro means, doesn't it?
I mean, somewhat. Does anyone actually recognize pyro anywhere else in this class? It's not the first time we've come up against the word pyro, or the prefix pyro. That's okay. We'll get to it. In fact, we'll start with it. So to begin, we'll kind of consider what was one of the first next generation sequencing. This is known as pyro sequencing. This is kind of a bit of a figure to demonstrate it. You all should have this figure in your notes. This can kind of give you a sense of this key. It's pretty nice. I got it from Wikipedia in this case. It was a pretty good little figure. So the first thing you do, step 1, get yourself a single-stranded DNA template and attach a primer. A primer in this case is where where a synthesis will begin. It is a matched sequence that will begin the synthesis. So step one, get yourself a single-stranded DNA template, jam a primer on it. We're pretty used to that at this point. Step two, go ahead and add some deoxynucleotides, all these little things that we've been playing around with, and then basically start to let our DNA polymerase, it could be Taq polymerase for instance, incorporate them into the template. And what this does, and this is what I was hoping someone would recognize, when that happens, we actually release pyrophosphate. This is what we had seen, I believe, in lecture 3. So if you want to look at your notes from lecture 3, you'll likely see that releasing of pyrophosphate during deoxynucleotide triphosphate incorporation, DNTPs incorporation, that gets released. Well, in pyrosequencing, what we do with that pyrophosphate is we use a thing called ATP sulfurase. I can't get that right to save my life. And this basically converts that pyrophosphate, PPI, to ATP, adenosine triphosphate. And then that adenosine triphosphate helps convert luciferin to oxyluciferin. Now, lucifer in this case is nothing to be afraid of. It's light, yeah, exactly. Just refers to the morning star. So by combining pyrophosphate with this ATP sulfurase, we release ATP, which then binds to luciferin, which then converts it to oxyluciferin, and that releases light. So all of this to say, every time you bind a DNTP, you get a little bit of light. And then basically the amount of light that gets incorporated, or that gets sort of shown, is proportional to the ATP. So if you have more ATP, more light is shown. So if you had done one sequence, another sequence, and another sequence, say they were all A's, well, now we've got a big spike in our A's. And then what we do is we detect that light with a camera, or a CCD. What's that, a coupled charge? I forget what the D stands for. And then gets analyzed. So basically you look for that light and you analyze it. And the graph kind of looks a bit like that over time. And then finally, step 5, this is kind of just parenthetically a step 5. All the unincorporated DNTPs and ATPs are degraded by a thing called ipyrase. I'm the worst at this. Ipyrase, which so they can be used again, and the whole cycle starts again. That is pyrosequencing in a nutshell. Any questions about that process to this point? And if you don't have them now, that's okay. You can add. Yeah.
Can you go back one just for a second?
Oops, I can go forward.
Yeah.
Yeah, not a problem. Yes.
So it tells, like the spike tells when it changes from one DNTP to another. But
Not when it changes from one DNTP for another. It means that we've incorporated a DNTP.
then how do you know which DNTP it incorporates?
You can control that. So what you can imagine is having, so there's a number of different ways you can do this, but say you've got just a channel that we just flow all our A's in at one time. We clear it all out, and then we flow in just our G's. Clear all that out, just flow in C's. That's one of the ways we can do it. There are other ways where you can use sort of a modified luciferin to react to particular kinds. That's a little more complicated than we'll deal with here. But in general, you do it one at a time. Yes? Step 2. So step 2, after we've got our primer there, we're just going to put it in a nice bath of DNTPs, in this case, a sequential bath, where we'll have A, D, ATP, D, CG, TP, and we'll flow those through, we'll incorporate them, and as we incorporate them, we'll release this pyrophosphate, which will be the basis of our reaction to generate light. Are you good? Yes.
So we know what it is based off of the time the light is released.
Exactly right. Yep. Exactly right. You all know this better than I do. Like I said, this is most famous for its implementation in a thing called Roche 454. There's actually a number of these on campus. You'll see them in lots of labs. These were actually discontinued in 2013. They're just not, with the next generation, they're just not that good at competing. So a lot of people, if you kind of look at my announcements every now and then, you'll see Roche's name come up a lot, and they're looking to see what their next move in this space will be, because a lot of people expect it to be big. Okay, so now our next one, single molecule real-time sequencing. As I said, single molecule real-time sequencing. And that actually looks a little bit like this. Sorry that the figure isn't the greatest. I really couldn't find much of a better one, and I couldn't really make much of a better one, but hopefully it suffices for you to understand what's going on. So, step 0. First thing you got to do before we can even do anything is have a whole bunch of fluorescent phospho-linked labeled nucleotides. And sort of that phospho link is going to matter in a bit. But what matters is we've got a bunch of nucleotides that have a little dye molecule attached. And they're attached at the phosphate group. So, before we've even done anything, make sure we got a bunch of these hanging around. Then, what you got to do, this is one of the neat tricks. This is a fabrication manufacturing process. What you got to somehow do is get your DNA polymerase affixed to the bottom of something. And this thing is called a zero-mode waveguide. And I'll explain that here in just half a second. Basically, what you got to do is take your DNA polymerase, rather than it being free out in the bath to do what it's got to do and incorporate nucleotides, you're going to jam it at the bottom of a well, and you're not going to let it leave. So that's the first thing you got to do. Now, what is this zero-mode waveguide, you may ask? A zero-mode waveguide with... Oh, did you want me to go back? No? I'm just trying to interpret hands and shocked looks at this point. Okay, a zero-mode waveguide is basically any kind of waveguide, an optical waveguide, wherein the volume is less than the wavelength of light. So light, as you know, or hopefully know, has an actual wavelength associated with it, usually in the 200, 400, 600 nanometer range. And so if you've got a volume that's smaller in all directions than the wavelength of light that you're going to use, it's called a zero-mode waveguide. And what a waveguide means is that you literally guide the waves. So a fiber optic can be a kind of waveguide because it's taking all those light waves, not letting it go anywhere else but out the front. So in this case, we are going to concentrate this and not let the light go really anywhere, right? We're going to shield it for being able to propagate out because we've made the volume so small. It's a neat little trick, and there's a lot of patents around it. Second step, and this is sort of the most important one, is that those fluorescent phospho-linked label nucleotides get incorporated by that DNA polymerase into the DNA, and this produces a little bit of a flash. Because now we've got all these phospholipids. We've got one that comes in down into the volume, and as the DNA polymerase plumps into place, it lights up the whole region very, very brightly. And that, if we look over time, sort of the signal intensity, we can see what that looks like. And this is actually real data. That's what it really looks like. Okay, step 3, we're going to cleave that phosphate chain and thereby release the molecule, the dye molecule. There's a number of ways you can do this. You can kind of modify your DNA polymerase to include a portion that'll cleave it. You can introduce new chemicals. You can introduce a new enzyme. You can introduce any number of things. But basically, we're just trying to get rid of that dye molecule because we no longer want it there. Because if it stays there, it's going to gum up our works, and it's going to drown out the next signal that's coming. Okay, and then basically, after that, we're just doing the steps again. So we incorporate a new one. In this case, it's thymine. It's going to produce another kind of flash. What we can do, instead of doing it sequentially, as we might do for pyrosequencing, we can actually just now start to use individually labeled nucleotides. And you can imagine how that might allow things to go faster, because now you can have a whole bath of these labeled nucleotides with their individual labels associated with them. So now we can, we don't have to do 4 runs for every one nucleotide we want to get. It greatly speeds up the process. And then step 3 again, cleave and repeat. So just keep incorporating, cleaving, and repeating, cleaving those molecules. And that is, in a nutshell, single molecule real-time sequencing. Any questions about that to this point? Again, if you have them, we can address them later. This is specifically the territory of a company known as Pacific Biosciences, often shortened to PacBio. These are their 2 famous machines. This is, I forget what it was called, it's like the PacBio RS2 or something. But because they had named it the RS2, they couldn't name this thing like the RS3. They decided to call it the sequel. So it's RS2-2, Electric Boogaloo. So yeah, this is PacBio, and I think they're out of Menlo Park, just to give you some sense of where these things are located. Menlo Park is in California, near the Bay Area, where a lot of this stuff is. Okay, the third technique we're going to talk about today is called ion torrent sequencing. And actually, it's, if you go, ion torrent is a brand name. It comes from Thermo Fisher. So when people try to speak of it generically, they say ion semiconductor sequencing. But it's kind of like in the field saying, I used a search engine to get the answer. It's like, oh, you Googled it. Yeah, I know what you mean. Oh, you blew your nose on tissue paper? No, it was Kleenex. So we're usually talking about ion torrent sequencing. And this is also pretty interesting. So recall that when we incorporate a DNTP, that we release a pyrophosphate. Well, one of the other things we release is actually a little hydrogen atom. And where pyrosequencing focused on that pyrophosphate, ion torrent sequencing is going to focus on the ion. This is just a little hydrogen. It's basically a protein, or not a protein, a proton. That's all it is. It's not basically that is what it is. So ion torrent sequencing is going to focus on it. And the way it's going to focus on it is it's going to use a thing called an ion-sensitive field effect transistor. And you don't need to necessarily know how this all works. This is a little beyond the scope of this class, unfortunately. It's, in fact, I'll be honest, beyond the scope of me, how it actually works. But in general, the way it works is the ion, basically when an ion comes into a bath that we've got some ionic solution. And when we start to add an extra hydrogen that occurs from our DNTP incorporation, this changes the ion concentration, which in turn changes the current that passes through the transistor. Does that make sense? We basically have some transistor whose ability to let current pass through it is determined by the presence of ions. The more ions there are, the more current flows through. This is a bit like, if you've ever heard the term a MOSFET. I should forget what that stands for. I really should have thought about that before putting it on a slide. But that's just another kind of chip, another kind of semiconductor chip made of silicon. I will look that up and make sure I mention it next class. And this actually looks a little bit like this. It's basically a bunch of tiny little micro-machined wells, usually micro-machined out of silicon. And behind it, there's an ion-sensitive layer. That's basically that little bath we were talking about. In this case, it doesn't need to be a bath. It can be any permeable membrane that can retain ions. And then we've got some proprietary sensors. These are just little things, little transistors that can measure the changes in each of the individual wells. So as we have a reaction occurring in each individual well, we can see as things get incorporated. This allows us to massively parallelize this thing. Any questions to this point? This one's probably a little the most confusing. Yeah.
A single hydrogen atom can change the current that much.
Well, so the hydrogen atom is changing the transistor, and the transistor itself can change the current. It's kind of like asking, this is one of those devil of a questions. If you were to actually think about the potential energy involved with me flicking on or off a light switch, well, all of my energy that I just flicked didn't actually get converted into turning it on or off. I just happened to be able to flip a switch that is able to do something else. So in that case, the transistor, I changed the property, in this case, of the transistor that's able to change the current flow without actually changing the current flow myself. Good question. Like I said, this is most primarily associated with Thermo Fisher. This is a little desktop machine. You'll also see these in a lot of hospitals, places where they do these sort of quick genetic screens. These are typically found there. All right, this one's the one we'll probably spend the most time with. This is usually, well, hopefully I didn't burn that one out, but the price of doing business. This is sequencing by synthesis. This is also known previously as Illumina dye sequencing. So if you kind of look in the literature, you might see one or the other. We're going to kind of hunker down on this one. When people talk about next generation sequencing, this is usually what they mean. Illumina kind of, their bread and butter looks a little bit like this. This is their latest and greatest machine. It's called the HiSiC-X, I think. And it's a big thing. It just came out in 2015. And with this machine, they're hoping to get to the sub $1,000 genome, which is crazy. I mean, crazy. Okay, so the steps of it are as follows. First step, you're basically going to take your whole genome or whatever thing of genetic material you want, and you're going to shake the heck out of it, and you're going to fragment it randomly. Usually you do it randomly. You can kind of, you can do it hierarchically, but Illumina just shakes the heck out of it and fragments it. Then, once you've fragmented it and split it into a bunch of different random pieces, you're basically, to each one of those random pieces, going to add an adapter. You're going to ligate an adapter to both ends of it. One will be a probe, or an anchor. The other will be a primer. This is where synthesis will begin. I don't have that written up here, so that's one of the things. Yes?
Can you define ligate again?
Ligate?
Yeah.
Bond. Yeah. I don't mean anything fancier than that. You're just going to take an adapter and jam it on the end. But I don't know, when we have to release these slides to ABET, I don't want to say, oh yeah, you just jam it in there. So I say ligate. Yeah, great question. Good clarification. But that's all of it. Then what you're going to do is you're going to bind each of those little single-stranded fragments somewhat randomly to just some flow cell surface. So you got some thing of glass or silicon, or I don't think there are any metal ones yet. Basically just some machined surface that also has corresponding little bound elements, little adapters themselves. You can kind of see we've got scattered little black ones and little gray ones, just like we have up here. And so they're all going to be bound, at just somewhat random. Well, once we've bound them to this surface, we're going to begin a process known as bridge amplification. This is a particular kind of PCR. We'll learn about other kinds of PCR, such as emulsion PCR, but today we're going to focus on bridge amplification. And this basically, the way you start it is something of a guarded secret and something of a chemistry nightmare. But basically, you just make the conditions just right. Remember we had talked about the Goldilocks condition? We got to make sure that in order to get these primers to match up to their little anchor points as they are here, we just got to make sure we get the conditions just right. The right temperature, the right pH concentration, ions and enzymes in their appropriate places, but we can basically fold those strands over. Once we've folded them over, we now just get to start polymerizing. We're just going to sick DNA polymerase along with some DNTPs, and we're going to synthesize the complementary strand. That's all that's going to happen. So in this case, like I said, we're going to add DNTPs, or dinucleotide triphosphates, and enzymes, most primarily DNA polymerase, to synthesize the complementary strand. Well, once we've done that, we're going to kind of shake it all up again, or heat it up a little bit, or we're going to basically agitate it enough so that we can plop off some of the, plop off one of the points, so that they're still anchored to the substrate, but not both ends are anchored. We're basically going to make them single-stranded again. And then we're just going to run through these steps again. And we're going to get big old clusters of them. We're going to just keep doing that until we got lots and lots and lots and lots of single-stranded DNA molecules, all kind of right near each other. You know, the fragments that are near each other are going to be more similar than those that are, you know, farther away. Typically. So we do that a whole bunch of times. Then what we're going to do, just like we did in Sanger sequencing, is we're going to add labeled nucleotides. And these are going to have a little fluorescent dye molecule on them. And we're going to take a picture of that little fluorescent dye molecule. But unlike Sanger sequencing, which terminates it and cuts off the whole thing, just like all the other techniques we had sort of discussed here, we're now going to be using reversibly terminatable. So things that can be, we don't have to terminate the sequence to send out our fluorescent signal. So we will bind a DNTP, we'll shine a laser at it, that'll cause it to excite within a certain region, and we will take a picture of that with a camera. We'll do that for each and every single base pair, and that'll look something like this. So in our first one, we've got a T, which we can see here, and the second one, we've got an A, which you can see here. This one, we've got a C. Yeah, I don't know, I guess. Oh, yeah, I was reading that backwards. Yeah, you're entirely right. It is, in fact, a G. And then finally, we've got a C here, which we see. So T, A, G, C. And even though we didn't do it for, or didn't show it for the other results, we can see that they also, and each one of these images, has a fluorescent molecule associated with them. Okay, so that is basically next generation sequencing. What this image actually, yes?
Can you go over step 7?
Step 7. So step 7 is just doing steps, I suppose that would be 6 and 5 over and over and over again. We're basically just going to keep them little single-stranded things that stand up, bend them down, do bridge amplification, stand them up, bend them down, do bridge amplification, over and over and over again. Yes?
Can you go back to 6?
We can do just that. So in this case, like I said, we're just going to denature the strands. We're going to separate them because in this case they're a double-stranded thing. We want them to be single-stranded. So we're going to just shake them or heat them or agitate them, you know, chemically potentially, to get them into single-stranded status. Yes? One of them is the anchor point, which has no polymerization. The other has a primer. And that primer just happens to also be on the surface. Because otherwise, how do you get the primer to the top of that thing repeatably, accurately, all the time? Great question. But you know, you can potentially change that and get yourself a patent. You know, like if you can do it different, you can potentially benefit. But yeah, that's a great question. Any others to this point? Otherwise, I'll just kind of step through. So this is sort of what that picture looks like. This is a pseudo color image. It doesn't actually look like this. They get excited in, you know, say non-visible realms. But what that allows us to do, you know, each of these colors is associated with a gene at one, or a base pair at any given point, and we can actually separate that out. So say A, G, C, T, and we can get a good sense of where they are all located at any given time. And we take these pictures over and over and over and over again. In fact, because we have 3 billion base pairs, we likely have to take a lot of these pictures. But if you're like me, it's a little confusing. How the heck does a picture like this actually turn into a genetic sequence? Well, this is going to be a bit of an example. And I'll release this example after class so that you can see it in our digest. Because you just will not be able to write this down. So just try to understand what's going on. So you can imagine, right now I'm just imagining we've got 16 spots where we're sequencing, where we're synthesizing. And in each of these I've just incorporated either a G, a T, an A, or a C that's giving off that fluorescent molecule that we care about. So in this particular case, I've now got G corresponding to G, T to C, C to C, G to G, and so on and so forth. Well, this is the first picture we take, and now we're going to take the next one. We've now sequenced the next base pair. This is what everyone's became. So now we've got G, T, G, A, A. Oh, we can take another picture, and now they've all kind of changed. And you can see we're starting to build out a sequence for each of these individual fragments. Does that make sense? Is that confusing?
How does that associate? I'm sorry.
What are you confused by?
Where are we getting all this?
So what I've done in Illumina is I've fragmented my DNA, and I got random fragments spread about this board. This is just, I had some, say, gene that I cared about. I diced it all up and stuck it on this silicon wafer. And now I'm synthesizing it, and these are the results we're getting. It's confusing still because even though we start adding tons and tons of these things, these base pairs, these nucleotides, we're not quite sure what to do with it. So we just keep getting a bunch of sequences. Well, now what we've got to do is a thing called alignment. There's two ways we can do alignment. We can either compare, which is I know what the gene is, and I know how to map something to it. So that is, you have a reference genome, for instance, the human genome. Or what you can do is a thing called de novo. You can align de novo. This is just where you correlate the fragments to one another. And that just means I've got, all of these things. How the heck did they all line up? And we're going to kind of skip the math for this for right now, but in this particular case, this example was a little easier. I'll show you sort of what I just did here. What I had done in this particular example is these last three actually match the first three. These last three match these first three, which means we have a bit of an overlap for each of these little fragments. They wouldn't normally come out that pretty, but this is a toy problem. So we can kind of overlay them one on top of the other to get what the whole gene itself is. Does anyone recognize that genetic sequence? Yeah, wow. Some of you actually do have that committed to memory, huh? That's great. Yes, that is actually your insulin A gene.
I just thought it looked similar. I was like, that kind of looks like it, huh?
You were able just to see, oh yeah, that TTTA, see, recognize that anywhere.
Yeah, I was like, yeah, that kind of reminds me of it. Maybe.
Yeah, so that's where I got it from. And this is how you can actually see where it took you and everyone in this class maybe a very long time to translate this whole thing. It would actually take you a pretty long time to write it all out. We were actually, with that 4x4 grid, in less than 7 steps, get a lot of coverage. We were able to actually sequence the entire thing really, really, really quickly. Yes?
Well, if you know how like the same places like have the same content overlap, so how do you like there's like that one has like
Great question. We're about to get into that. So yeah, you're already anticipating. I like where your mind is at. Yes?
I know that grid is a simplification, but how do they turn all those dots to something? I don't know. I don't understand how it translates from that picture of just a bunch of dots into that grid.
So each one of those dots is itself a genetic a base pair. It's either an A, a G, a C, or a T. So if it was colored yellow there, it was maybe an A. If it was colored green, it was a G. And so I know where they all are on my grid. And so if I just start to take a picture and at one point it's green, and then the next time I take the picture it's yellow and the next time it's blue and the next time it's red, I now know it did say, you know, A, T, C, T. Does that?
They all overlap, like the exact same spot on one.
Assuming it changes over time. So in this case, one picture to the next would just give you the same color. But over time, yeah, the same spot would change color. Yes?
You mentioned that the process is pretty fast. How fast is it to go through the steps?
We will get into that, but it's on the order of milliseconds, and even microseconds is what they're targeting. So it goes very fast. At the longest point, it will take, so with their first one, I think it took 1 to 3 seconds, depending. Now they're down to millisecond range. Yes?
Can I ask how many pictures do you take, like, per second?
A lot.
Okay.
It is not uncommon for a next generation sequencing machine. So if you were to actually sequence a human genome, say we take it from your swab, and rather than PCRing it and gel extrophoresising, stuck it in an Illumina sequencer, it would generate tens to hundreds of gigabytes to terabytes. It's not uncommon for it to be like a 10 terabyte data file. We're in the era of big data. So this little bit of overlap is actually called your depth, or your coverage. And in this particular case, we've got, say, 3 nucleotides that have a depth of 2. So we've got 1, 2. That means it is the number of overlapping sequences you have for a given nucleotide at a given base pair. So let's just see another example of that. I did another 4 experiments when you weren't looking, and now we've got a depth of coverage of 3 in this case. We've got 1, 2, 3 for all 3 of those. Now you'll actually see a couple of these. These 2, I suppose, or I guess just that one, just has a depth of coverage of 2. Does that make sense? Because we've only got, for this G, we've got 1, 2, but there's no third one down. Well, just like that, when you blinked your eye, I ran another experiment, and now we've got a depth of coverage of 4 for these for this particular sequence. Here we've got a depth of coverage of 3 around those G's and those T's. So that's known as depth and coverage. Now we're actually going to get to your question. So here's what some actual data looks like. I don't want you to be afraid of it, but this is what it looks like. And actually, only kind of. Like I said, you can download this stuff from the internet. You can do this. So how do we actually determine where something goes, where a read goes? Because if you look at it, we got a lot of sequences that look pretty darn close to one another. So if I had this sequence way up here, and I said, where does it fit in? Does it fit in here, or does it fit in here? Well, we want to test those hypotheses. In this particular case, our red sequence, or read, the sequence that we read, our red sequence, not the color, we're trying to match it to our reference. Well, it's got a couple of extra base pairs. It's got a couple of differences that we can see, and there's actually a pretty big gap. Well, in our second hypothesis, well, there's maybe 1 or 2 base pairs that aren't quite right, and we got one little gap in comparison to the reference. But which of these would you suspect is the correct place, the more correct place to place it? Yeah, probably hypothesis 2. And so even though we've got a few errors, it actually makes more sense than hypothesis 1. We can chalk that up to things like sequencing errors, or synthesis errors, or our reference not being quite what we need. Because say you've got someone's human genome, it's actually not going to match your genome, no matter how many times you try. Okay, but why are there still gaps? So if you actually look here, we've got a couple of gaps. And here I'll kind of show you that at different, no. So why are there gaps? I can answer this by just saying, these machines actually have tons and tons of errors. The error rate is actually about 1 in 1,000 to 1 in 10,000. Well, that's pretty good. That's 99.89% accurate. It is still enough to cause problems. But we can, while we're running these correlative maths, just punish the gaps. Okay, but what if we actually come up against a tie? Say we've got two sequences, so I've got that same read sequence up here, and I've got one example where I've got one base pair different, and I got another example where I got another base pair difference. How do I break this tie? How do I know where it actually fits? It seems to fit in both places. Well, there's actually a thing that the sequencer is going to try to do called a quality measure. Like I said, real data. And so you don't just get, as I said, all these little base pairs. You also get from the sequencer some measure of its quality, how good it feels its guess is at the base pair. Because that machine is going to try to say, I got that one, I nailed that one. That's 100%, that's as good as I'll ever do. Maybe sometimes it's 50% because the signal gets muddy or there just wasn't anything incorporated. So there's a thing known as fast Q format. You don't necessarily need to remember that, or memorize it rather, but you should be aware of it. And fast Q format basically allows us to code in little ASCII characters things such as B or the equal sign or the percentage. You know, as we see here, B equal percentage. And these represent integer values for our quality measure Q. So we've got some quality measure, and the sequencer is going to try to take some stab at what it's supposed to be. In this particular case, our quality Q is defined as -10 log base 10 of P, where P is the probability that the machine called the wrong base. So that means if we've got a Q of 10, our chances of getting something, of getting the base wrong are 1 in 100, or 1 in 10. If our Q is 20, our chances of getting the wrong base are 1 in 100. If our Q is 30, it's 1 in 1,000, and so on and so forth. And you can kind of see from that probability how that would, log base 1,000 times 3. And so the higher the better. So now, if we had sort of two sequences, our first sequence where our Q is equal to 30, and the second hypothesis where our Q is equal to 10, what is the better placement? Who thinks it's hypothesis 1? Who thinks it's hypothesis 2? Who doesn't believe in voting when your professor asks? Oh, really? Okay. Well, it turns out you're the ones that said hypothesis 1. Most correct. Because our Q value is the best. The higher the better. Okay, sequencing by ligation. Yes, actually, ask me after class. Sequencing by ligation. This is the finest thing we'll talk about, and in fact, the only thing I'll say is it's basically sequencing by synthesis, except now we're doing it on more than one base pair at a time. So every time we take a picture, it's not just one base pair we're capturing, we're actually capturing a bunch of base pairs all at once. Give you some sense of what that looks like, where synthesis, or sequencing by synthesis, we just keep placing them one at a time, where we've got ourselves a little dye molecule, we've got our DNTP here. Now what we've got is we've got some big old primer, some just thing, of multiple base pairs. In this case it's 5, but it could be 2, it could be 20, it could be however many you want. And it's also got a unique fluorescent molecule. What this allows you to do is come up with little template strands that allow you to constantly bind them so that you can do, say, 5 at a time. Because if you happen to know that this molecule is associated with this strand, then now you can do it 5 at a time rather than doing it 1 by 1 by 1 by 1. So that's synthesis by, synthesis. That is sequencing by ligation. Okay, so in summary, I've given you a little table here. And if you haven't, you can go ahead and check it out online. So the first thing, we've got pyro sequencing. The average read length here is about 700 base pairs. It's a pretty darn accurate thing, and it can basically do 1 million runs per, or 1 million reads per run, and it takes about 24 hours to do a whole genome, and it costs about $10 per base. So you can do it fast, but it's pretty expensive. Sequencing by, or single SMRT is the least accurate of all the things we just talked about, but it's very, very fast, and it's pretty cheap. So that's one of the reasons you'd use it. And again, you can kind of consult this yourself. Our ion torrent sequencing, very accurate, very fast, a little expensive. And especially there's a thing we didn't quite get into, but it's very susceptible to things called homopolymer errors. So if you have a lot of T's in a row, it can lead to some problems. Sequencing by synthesis by Illumina is probably the gold standard. It's the cheapest. It's often the fastest, but not really. It's somewhat slow, very, very cheap, and gets very deep coverage, which is what makes it very accurate. And finally, sequence by ligation, pretty slow, but cheap, still good. All this to say, I want to remind you that in about 1 to 2 weeks, the test looms. I will be releasing your homework by Wednesday, and you'll have a week to do it after that. So as the test looms, the struggle resumes, and I will see you all next time.